I want you to imagine sitting in a deserted church late at night.
Okay, setting a scene right away.
Yeah, well, just picture it. It's completely dark, it's entirely quiet, and it feels, profoundly serene. You're just alone with your thoughts.
Right.
But the truth is, in that exact moment of absolute peace, you are actually being bombarded.
Oh, completely.
You are sitting in the middle of this massive, chaotic, informational tapestry. I mean, dim light from the altar is crashing into your eyes. The microscopic sounds of the city outside are vibrating through the walls into your ears.
Mm-hmm.
Even the hardwood of the pew is actively compressing the nerve endings in your skin, and the complex chemical makeup of incense is drifting into your nasal passages.
And that's just the stuff you actually have the biological hardware to process.
Exactly. Because you're also being bathed in radio waves and gamma rays, magnetic fields,
Yeah.
maybe even a dog whistle from the street outside.
Yeah, it's total sensory chaos, but you only experience the serene quiet.
It's wild to think about.
It is a beautiful image, though, and it captures the central paradox of our biology perfectly. We walk around feeling like we are experiencing the complete objective reality of the world.
Right, like we're seeing everything there is.
Exactly. But we are actually just filtering it. I mean, if we look at this from an evolutionary perspective, human beings only evolved to detect what helps us survive.
The essentials.
Yeah, we need to know where a piece of fruit is, what is a safe path to walk, and, what represents a predator. If we possess the biological hardware to process every single radio wave or X-ray bouncing around that church,
We'd short circuit.
our nervous systems would be instantly and permanently overwhelmed. We are quite literally designed to ignore almost everything in the universe.
And that is the mission of our deep dive today. We are going to look at how our bodies act as these highly specialized, incredibly selective input systems.
It's a fascinating topic.
We are going to explore the absolute biological limits of human sensation, the messy psychology behind how our brains decide what is a true signal and what is just noise.
Which is trickier than it sounds.
Oh, absolutely. And we'll look at the sheer mechanical genius of our eyes and ears. And finally, some incredible new research on how the brain literally alters reality before we even consciously perceive it.
So, to really understand how you experience the world, we first have to establish what your body is physically capable of detecting. We have to find the absolute floor of human sensation.
Okay, let's unpack this because the absolute thresholds of human sensation are just wild.
Mm-hmm.
I think a lot of us assume humans are kind of blunt instruments compared to, say, an eagle with its vision or a bloodhound with its nose.
Sure, yeah.
But the numbers are staggering. Under the right conditions, you can see a single candle flame from 30 miles away on a dark, clear night.
30 miles, it's crazy.
And you can smell one single drop of perfume diffused into a volume of space the size of a 6-room apartment. You can even feel the wing of a fly falling on your cheek from just 1 centimeter away.
Right, and those benchmarks represent the absolute threshold.
Yeah.
By definition, that is the minimum magnitude of a stimulus. You can reliably discriminate from absolutely nothing at all.
Right.
But to prove just how insanely sensitive our biology truly is, we have to look at this classic 1942 experiment from Columbia University.
Oh, the Heft experiment.
Yes, led by a researcher named Hecht. They wanted to find the absolute threshold for human vision, but not using a candle. They used the smallest physical unit of light in the universe.
The photon.
Exactly.
A photon.
I love this experiment so much. They put people in a completely dark room, let their eyes adjust, and then fired unimaginably tiny flashes of light at them to see what they could actually detect.
Mm-hmm.
And the researchers found that a person can detect a flash of light containing just 100 individual photons. But what blew my mind was what happens once those photons actually cross the threshold of the eye. Well, yeah, because the physical journey into the eye is treacherous for a photon. Out of those 100 photons entering the pupil, only about 7 actually survive the journey through the fluids of the eye to reach the critical molecules in your visual receptors.
7.
Yeah, the rest are just absorbed or scattered by other tissues.
So if only 7 photons are hitting the receptors and you can still see the flash, that means your eye is triggering based on almost nothing.
It goes even further, actually. Because those 7 photons are scattered, they land on 7 different receptors.
Really?
Yes. That means the critical receptive unit of the eye, a single biological molecule, is being activated by one single photon. Wow. Human vision is literally as sensitive as is physically possible in our universe. You cannot detect less than one photon.
Yeah.
Biology has pushed our visual hardware to the absolute quantum limit.
That is just incredible. But sensing that something exists is just part of the equation, right? We don't just stare at the static universe.
No, we are constantly moving, and the environment is constantly shifting. We have to detect change. And that requires a totally different mechanism. This brings in the difference threshold, or what psychologists call the just noticeable difference. Like, how much does a stimulus need to change before your brain registers that a change has occurred?
And this operates on a principle known as the Weber-Fechner law,
Yeah,
right?
exactly. The core idea is that noticing a change isn't about the absolute amount of change. It is entirely proportional to the original intensity.
Okay, so if you are sitting in a room lit by 25 candles and someone quietly lights 2 extra candles, your brain registers the room getting brighter. That is an 8% increase in light.
But, and this is the key, if you were in a massive hall lit by 100 candles and someone adds those same 2 candles, you won't notice a thing.
Because the proportion is off.
Right. The physical change in light is exactly the same. 2 candles worth of photons, but your perception ignores it. To trigger your conscious awareness in the 100-candle room, you would need to add 8 candles to hit that same 8% proportional threshold.
So it scales, which flows right into how physical intensity translates to psychological intensity, something called Stevens' power law.
Yes, Stevens' power law.
Let me try an analogy here to see if I have this right. It seems like harmless things, like the brightness of a room, operate on a curve that bends downward.
Mm-hmm.
It's like a biological volume knob that actively limits how loud your psychological experience gets. It compresses the signal to prevent sensory overload.
That's a great way to put it.
But dangerous things, like an electric shock, operate on a curve that bends sharply upward. So a tiny physical increase in the voltage of a shock creates a massive screening spike in your psychological perception of pain.
Let's take that volume knob analogy a step further because it perfectly illustrates our evolutionary alarm system. If a light gets incrementally brighter, that is just data. Your brain turns the volume down so you can still function. But if you touch a hot stove, your sensory modality cannot afford to compress that signal. It needs to scream at you immediately. It curves the perception of pain upward exponentially so that you violently pull your hand away before your tissues literally burn.
So your perception is being actively manipulated to protect your physical body.
Exactly.
But a hot stove or a candle in a dark room, and these are very clear, distinct signals. The real world isn't a controlled lab experiment. The real world is messy, it's chaotic, and our senses are constantly flooded.
They really are.
So how does the brain actually decide what is the true signal and what is just random background noise?
Well, that introduces the psychology of signal detection theory. In the real world, you are rarely just looking at a clean signal. You are hunting for a meaningful pattern buried under a mountain of noise.
Right.
Take the field of medicine. An X-ray is not a pristine, high-definition photograph. It is a shadowy, overlapping grayscale mess of tissue, fluid, and bone.
There is a medical malpractice case from our sources that perfectly illustrates this trap.
Oh, the radiologist lawsuit.
Yeah. So a radiologist looks at a patient's X-ray, clears them, and sends them home. Years later, the patient tragically passes away from a tumor that was present, but very faint, on that original X-ray.
Right.
The family sues. They bring in an expert witness, another doctor, who looks at that original years-old X-ray and confidently states, the tumor is right there. It is obvious. The first doctor was negligent.
And it feels like an open and shut case of negligence, right? But human perception is far more complicated. In signal detection, we categorize outcomes into hits, misses, and false alarms.
Okay.
The first doctor missed the faint signal, but when evaluating the expert witness, we have to completely separate his physical sensitivity to visual data from his psychological expectation.
Because the expert witness already knew the outcome.
Exactly. The expert doctor already knew the patient died of cancer. That knowledge completely changed his psychological bias.
Wow.
He wasn't necessarily possessing better, more sensitive eyesight than the first doctor. His expectation just drastically lowered his threshold for saying, yes, that ambiguous gray smudge is a tumor.
So in the terminology of the theory, the first doctor was like conservative Charlotte, who needs a lot of evidence to say yes, and the expert was liberal Linda, who says yes to almost anything that looks like a signal.
That is exactly the framework, bias versus sensitivity.
Wait, so let me push back on this on behalf of anyone listening. We all have that one friend who claims to have Incredible observation skills, right?
Oh, sure.
They spot every tiny background detail in a movie, or they notice a weird noise in the car engine before anyone else. Are you saying someone who claims to have incredible senses might not actually have better vision or hearing?
Real.
They might just be wildly biased, like they are just a liberal Linda constantly raising false alarms.
It is highly probable, yes. They might just be guessing yes a lot. If you are highly motivated to find patterns, you will constantly point out faint signals. You will get a lot of hits, which makes you look incredibly observant. But without measuring how many false alarms they make, how many times they point out a noise that turns out to be nothing, you have no idea if their actual sensory hardware is superior to yours.
That makes total sense.
They might just have an incredibly liberal bias for declaring that a signal exists.
That completely reframes how I think about observation. It's not just the camera. It's the software processing the image.
Exactly.
But if our brain's expectations can trick us into seeing things that aren't there or missing things that are, how reliable is the physical camera feeding us that data in the first place?
Well, the human eye is arguably the most complex physical signal processor we possess. And the brilliance of the eye is that it doesn't just use one camera. It operates using two entirely different cellular systems housed on the exact same retina.
These are the rods and the cones.
Yes.
And they divide the labor completely. Cones are built for daytime driving. They handle color and sharp, high-resolution details. And to get that sharpness, they are packed tightly into the very center of your retina, an area called the fovea. And the wiring is VIP level. Each cone gets its own direct, one-to-one neural connection to the brain.
It's like having a dedicated high-speed internet cable for every pixel, which ensures you see incredibly sharp details.
But then you have the rods.
Rods, on the other hand, are designed for the night. They do not process color, they only see the world in grayscale, and they are spread out around the periphery of your retina.
So they're on the edges.
Right. Because they are designed for survival in low light, they pool their resources. You might have 50 rods sharing a single neural connection to the brain.
Oh, wow.
This pooling sacrifices sharp resolution, but it massively increases their sensitivity to the faintest hints of light.
Which gives us an incredibly practical trick. If you're ever outside at night looking up at the sky and you want to see a very dim, faint star, do not look directly at it.
No, you won't see it.
Right. If you look directly at it, you are pointing your fovea at the star. But your fovea is full of cones, and cones are useless in the dark. The star will literally vanish from your vision.
It's a great party trick.
Instead, you have to look slightly to the side of the star. That projects the faint light away from the center and onto the outer edges of your retina where all those highly sensitive rods are waiting to catch it.
And those rods require patience, by the way. If you walk from a bright sunny afternoon into a dark movie theater, you are temporarily blind.
We've all been there.
Your vision undergoes a two-stage process called dark adaptation. Your cones try to adapt first, but they max out their sensitivity in about 5 minutes. It's not enough.
So the rods have to kick in.
Exactly. And your rods take over, but they require a full 30 minutes in the dark to reach their maximum sensitivity.
Okay, so the rods and the cones are gathering the raw photons, acting like pixels. But wait, earlier we talked about how the brain edits things. Does the eye just pass this raw pixel data directly to the brain, or is it already messing with the image?
Oh, the eye is actively editing the image before the signal even reaches the optic nerve.
Really?
Yeah, it uses a biological trick called lateral inhibition to physically enhance edges and contrast.
How did that work?
Imagine a grid drawn on a piece of paper. Solid black squares separated by thick, bright white lines. It's known as the Hermann grid. If you look at this image, your brain will produce ghostly gray smudges right at the intersections where the white lines cross.
I've seen this. It's completely trippy. As soon as you look directly at one of those gray smudges to focus on it, it vanishes. Why does the eye invent a gray smudge that isn't there?
It comes down to how the cells are wired to talk to each other. A cell looking at that bright white intersection is receiving blinding light, but it is also receiving signals from the bright white lines above, below, left, and right of it.
Okay.
Those neighboring cells are wired to actively inhibit or turn down the volume on the firing rate of the center cell.
Wait, they tell it to quiet down.
Yes. Because that center cell is being aggressively told to quiet down by its neighbors on all four sides, the signal it sends to the brain is weaker. A weaker signal translates to darker. Hence, you perceive a gray smudge.
So the eye is literally photoshopping the image to exaggerate contrast so we can clearly see the edges of objects.
Actually, yes.
And this biological wiring gets even more fascinating when we look at how we perceive color. Historically, There was a massive, decades-long scientific war over how human color vision actually works.
It was quite the debate.
On one side, you had the young Helmholtz trichromatic theory. They argued that we only have three specific types of cones, one tuned to short wavelengths for blue, one for medium wavelengths like green, and one for long wavelengths like red.
Right.
And they said every single color in the spectrum we perceive is just a blended mixture of those three cones firing in different ratios.
It makes logical sense, and it is exactly how modern television screens work, mixing red, green, and blue pixels. But another scientist, Ewald Hering, pointed out that this theory completely fails to explain human experience.
He had a different idea.
He introduced the opponent process theory based on a few weird phenomenological facts. For instance, you can easily picture a yellowish green. You can picture a bluish red, like purple.
Sure.
But try to picture a reddish green or a yellowish blue.
You can't.
You can't. They simply do not exist in human perception. Hering argued that color isn't a mixture. It is processed in opposing pairs, red versus green and blue versus yellow.
So who won the war? Are we mixing three colors or are we processing opposing pairs?
What's fascinating here is that modern biology proves them both perfectly correct.
Really? Both?
Yes, it is a two-stage filtration system. At the level of the retina, the trichromatic theory wins out. You physically only have three types of cones: red, green, and blue.
Okay.
But as those electrical signals leave the eye and travel up into a neural relay station in the brain called the thalamus, the architecture changes. Those three cone signals feed into opponent cells.
Okay, let's unpack this. How does an opponent cell physically stop me from seeing reddish-green?
Think of an opponent cell like a literal seesaw. The red signals push one side of the seesaw up, causing the cell to increase its firing rate. The green signals are wired to push the other side of the seesaw down, decreasing the firing rate.
Oh, I see.
Because a single physical cell cannot possibly increase and decrease its firing rate at the exact same millisecond, you can never perceive red and green in the same spatial location. The biology fundamentally prevents the perception.
The mechanical constraints of the body dictating our reality. I love that.
It's elegant.
Now, vision is entirely based on absorbing electromagnetic energy. But hearing operates on a completely different physical plane. Hearing isn't about energy absorption. It's entirely mechanical. It is a physical symphony of vibrations taking place inside your skull.
Right. Consider what a sound wave actually is. It is just a physical change in air pressure, molecules of air bumping into each other.
Like a domino effect.
Exactly. The frequency of those waves, how fast they are hitting you, determines whether you hear a high pitch or a low pitch. And the amplitude, how intensely they are hitting you, determines the loudness, which we measure in decibels.
And when those invisible pressure waves crash into the side of your head, the mechanical architecture of the ear has to convert air movement into an electrical brain signal.
Which is a crazy process.
It is. First, the wave travels down your ear canal and physically strikes your eardrum, vibrating it like the skid of a drum.
Oh.
But behind the eardrum are three microscopic bones, the smallest bones in the human body, acting as an intricate lever system.
The malleus, infiscus, and stapes. And those bones are absolutely vital because the actual sensors of the inner ear are not floating in the air. They are submerged in a fluid-filled, snail-shaped tube called the cochlea.
Right, they're underwater, basically.
Yes. If sound waves in the air just hit that dense fluid directly, they would bounce right off. You need a mechanical bridge to overcome that density. Those three tiny bones act as mechanical levers, taking the broad, weak vibration of the eardrum and concentrating it into a powerful physical punch against the fluid of the cochlea.
And that punch sends a physical wave rippling through the fluid, which bends microscopic hair cells lining the tube. And the physical bending of those hairs is the spark.
Exactly.
That is what finally triggers the electrical nerve impulse to the brain. It is a stunning Rube Goldberg-level mechanical chain.
But precisely because it relies on tiny, delicate moving parts, it is highly vulnerable to physical damage.
Like with rock musicians.
Exactly. Rock musicians like Pete Townsend from The Who frequently suffer from sensory neural hearing loss. When you expose yourself to 120 decibels of sound consistently, the sheer physical force of those fluid waves violently bends those delicate hair cells until they snap and break.
And they don't grow back.
No.
Wow.
Unlike cells in your skin, they do not regenerate.
So we have this fluid-filled tube vibrating. But how does this mechanical system actually allow us to perceive different pitches? Does the ear just act like a microphone membrane, where if a high-pitched 1,000 hertz tone hits the ear, the whole internal membrane just shakes 1,000 times a second?
For a long time, scientists assumed it worked exactly like a microphone. This was called the temporal theory. But there is a massive biological bottleneck.
The speed limit of nerves.
Right. Human nerve fibers have a speed limit. They max out at firing about 1,000 impulses per second. But humans can easily hear high-pitched dog whistles at 15,000 or 20,000 hertz. The nerve literally cannot fire fast enough to match the frequency of the sound wave.
So the microphone analogy breaks down completely.
It does. This led to a breakthrough called the place theory. Researchers discovered that the membrane inside the cochlea doesn't vibrate uniformly like a drumhead. It moves more like a bedsheet when you grab the edges and snap it.
Oh, that makes sense.
Depending on the frequency of the sound, the wave will peak and crash at a different specific physical location along the membrane. High pitches crash at the very front now. Low pitches crash at the very back.
So your brain knows what pitch you are hearing simply by determining which physical neighborhood of hair cells is currently being bent.
Precisely.
So, just like with color vision, our bodies use a hybrid system. We use the temporal theory microphone trick for low, rumbling pitches, and we switch to the place theory bedsheet trick for high pitches, seamlessly stitching them together to cover the entire audible spectrum.
The integration is flawless, but as complex as vision and hearing are, they are ultimately remote sensors. They map the world from a safe distance.
Right.
When we transition to smell and taste, we are dealing with our chemical senses. They require intimate, physical contact with molecules. They're ancient and they feel incredibly primitive.
Primitive is the perfect word. Smell, or olfaction, gets a VIP bypass route in the human brain. Unlike sight or hearing, which have to stop at various neural relay stations to be processed and filtered, the nerves from your nasal cavity plunge directly into the olfactory bulb.
It is an unfiltered pipeline straight to the most ancient emotional centers of the brain.
And you can clearly see how ancient this chemical wiring is when you observe animal behavior, particularly how insects rely on pheromones.
Oh, absolutely.
There is this incredible experiment with ants that perfectly illustrates this. When an ant dies, its body immediately begins releasing a specific chemical compound, essentially the smell of death.
Right.
When the other ants in the colony detect this chemical, they react robotically. They pick up the dead body, carry it out of the nest, and dump it in the colony's refuse heap. So researchers decided to test the power of this chemical cue. They extracted the smell of death and sprayed it onto a living, perfectly healthy, moving ant.
The result is almost comical, but deeply profound.
It is. The other ants immediately grabbed their living, squirming friend, carried him out, and threw him in the graveyard. The live ant would walk back into the nest, and they would immediately grab him and throw him out again, over and over.
Wow.
They could see him moving. They could feel him fighting back. But the chemical smell of death completely hijacked their behavior. The smell overrode all other sensory data.
It's a powerful demonstration of how chemical communication dictates reality. And while human beings love to believe our massive cerebral cortex elevates us above such primitive programming, we are far from immune.
We really are.
In one fascinating study, researchers had participants wear undershirts for 24 hours straight without using deodorant or showering. Afterwards, participants were blindfolded and given 3 shirts to smell their own, a stranger male's and a stranger female's.
And relying purely on odor with no visual cues, the vast majority of people could correctly sniff out their own worn shirt.
Yes.
And they could accurately identify the gender of the strangers who wore the other 2 shirts. We are basically just mammals in clothes, constantly giving off and reading invisible chemical cues.
And our sense of smell is inextricably linked to our sense of taste, or gustation. Taste evolved as a primitive chemical sorting system for survival. We have 4 basic taste qualities, each with a clear evolutionary purpose.
Sweet, sour, salty, bitter.
Right. Sweetness signals energy-rich carbohydrates. Sourness acts as a warning for spoiled food. Saltiness indicates essential minerals for nerve function. And bitterness is a massive biological red flag for poisonous plant alkaloids.
We've all seen that tongue map from grade school, right? Where the front of the tongue tastes sweet, the sides taste sour, and the very back is a dedicated zone for bitter to stop you right before you swallow poison.
The catch is that modern research shows that MAP is an oversimplification. While certain areas have slight sensitivities to specific tastes, Every single taste bud on your tongue is capable of detecting all four taste qualities.
Oh.
More importantly, those taste nerves actively talk to each other and inhibit one another. And the final perception of whether a taste is good or bad is heavily modified by cultural training.
For example,
Well, communities in the Karnataka province of India consume diets rich in extremely sour and bitter foods. Their brains have been culturally trained to find the sharp bitterness of quinine to be highly pleasant, whereas a typical Western palate would aggressively reject it.
here's where it gets really interesting, though. What we colloquially call the flavor of a beautifully cooked meal isn't actually happening on your tongue at all.
Not at all.
When you get a severe cold and you complain that you've lost your sense of taste, you actually haven't. If I gave you a spoonful of salty broth, your tongue would easily detect the salt. What you've lost is your sense of smell.
Right.
As you chew, chemical vapors travel up the back of your throat into your nasal cavity. Flavor is a magnificent unified illusion created by the brain dynamically blending basic taste data with complex olfactory smells.
Which brings us to the ultimate question of this entire journey. Where exactly does sensation end and perception begin? We've tracked photons hitting the retina, pressure waves shaking the cochlea, and chemicals binding to receptors.
But where does the raw data stop and the brain's interpretation take over?
To answer that, we have to look at some cutting-edge fMRI research using a classic visual illusion.
The 3D sphere illusion.
Yes. Picture a drawing of a 3D hallway stretching out in front of you. Placed in this hallway are two identical checkered spheres. One is drawn near the bottom of the page, so it looks like it is resting close to your feet.
Right.
The other is drawn near the center of the page, so it looks like it is resting far away at the end of the hallway, even though both spheres take up the exact same physical amount of ink on the page, and therefore cast the exact same physical size of an image onto your retina. Your brain refuses to see them as equal.
Right. The sphere that appears further back looks massive compared to the front one.
The psychology behind the illusion is well understood. Because the context clues of the hallway tell your brain the second sphere is far away, the brain calculates that it must be physically gigantic in reality to cast an image of that size all the way to your eye. The brain scales the perception up.
But what functional MRI scans recently revealed about where this happens is truly groundbreaking. You would assume this complex 3D contextual math happens high up in the brain's cognitive processing centers where we think and reason.
Exactly. But the scans showed that this physical scaling happens in the primary visual cortex, known as V1. This is the very first, most fundamental stop for visual information as it enters the brain.
So the V1 region actually recruits and allocates a literally larger physical area of brain tissue to map the sphere that your brain thinks is bigger, even though the raw photon data coming from the eye is physically identical for both spheres.
This raises an important question. What exactly is reality? Your brain is literally overwriting the raw physical data from your eye to force a 3D reality onto your perception at the very earliest possible biological stage.
Before you even know it's happening.
Exactly. The map of the world is being fundamentally altered and rewritten before the conscious part of your brain even has a fraction of a millisecond to think about it. Interestingly, the research shows that people who physically possess smaller V1 brain areas actually experience a much stronger, more exaggerated version of this visual illusion.
The hardware dictates the software.
Yeah.
It's wild.
It really is.
So let's bring all of this together. We started in that quiet, dark church. We learned that to see a single flicker of light, your eye operates at the absolute quantum limit, requiring just one photon per molecule.
Incredible sensitivity.
We learned how microscopic bones act as mechanical levers to punch sound waves into fluid.
Yeah.
We saw how ants will blindly carry their living, struggling friends to a graveyard based entirely on a chemical scent.
All right.
And finally, we discovered that our visual cortex is actively editing and redrawing reality on the fly to force the chaotic universe to make logical sense.
It perfectly highlights that our bodies are not just passive open windows to the world. They are highly active, highly evolved, and heavily guarded filtration systems, picking out only the exact signals we need to survive and aggressively ignoring the rest.
So we leave you with this final thought. If our incredibly complex sensory hardware only picks up a tiny, highly curated fraction of the universe, completely ignoring the radio waves, the magnetic fields, the gamma rays, and the microscopic world, what entirely different dimensions of reality are happening right next to you, right now, in the room you are sitting in, that you simply do not have the biological hardware to feel.
Something to think about.
It makes you wonder what you're missing. Until next time, thanks for taking the deep dive with us.